Hey everyone, so these are the notes for the mega sheet that we didn't get to do in class. Take your mega sheet and it should be folded now into 4 squares just like we always do so that it fits in your notebook. And what we're going to do is a review / introduction to nucleic acids. So back in Unit 1 we talked about nucleic acids. We talked about their structure, CHONP. We talked about the bond being phosphodiester. We talked about their function, which is the genetic material of the cell, and that's what we're going to get into in this class, or in this unit. And the biggest thing is that we are looking at the structure of the nucleotide. So with the nucleotide, we're going to go into detail about that. Nucleotides are made up of 3 things: a 5-carbon sugar, a nitrogenous base, and a phosphate group. And this is what we're going to do over here off to the side. We're going to get into the nitty-gritty details because you need to understand the structure of that nucleotide in order to understand the difference between the lagging and the leading strand when we're talking about DNA replication, which is on your flipped video. So I'm going to draw the structure of this nucleotide starting with that 5-carbon sugar. We'll start with an oxygen group, and it's going to look like a pentagon shape. Remember that each corner represents a carbon, except for this is not a corner, that's our oxygen group. So when we number this, it's very important that you follow the same convention when we're doing any of the nucleic acids. So starting with this carbon over here, this is carbon number 1, carbon number 2, carbon number 3, carbon number 4, and this is carbon number 5. So you start with the one to the right of the oxygen and you're going to go clockwise 1, 2, 3, 4, 5. That is the 5-carbon sugar. You want to be on the lookout for carbon number 2. If there is an OH group, that is deoxyribose. I'm sorry, that is ribose. And if that OH group at carbon number 2 is missing, then it is deoxyribose. So that's a little @ sign. So if there's an OH group at carbon number 2, then it's ribose. If that OH group is missing, then it's deoxyribose. That nitrogenous base is going to be attached to carbon number 1. And we've talked about the nitrogenous bases being either your purines or your pyrimidines. And then that phosphate group is attached at carbon number 5. So understanding the structure of this nucleotide is very important to understanding the structure or the way that DNA replicates. It's going to factor in. I'm not drawing all the OH groups and H groups that would be possible on this 5-carbon sugar. We're just focusing on the ones that you need to pay a little more extra close attention to. So that's it for this box right here. Underneath it, we're going to take this square, or rectangle I should say. We're going to divide it in half. So that's right along the crease. And now I'm going to just divide this one in half again. And then what I'm going to do is summarize the history of DNA that we have been working on so far, starting with your Hershey and Chase. These 2 scientists were the ones that proved that DNA was the genetic material. We also talked about Meselson and Stahl. These are the ones that we looked at, we used for their experiment for semi-conservative replication. We discussed Watson and Crick. about the structure, describing the structure of DNA as that double helix. But remember that with Watson and Crick, we were using Franklin's crystallography, X-ray crystallography, for that structure. So we are going to talk about a few more scientists, so leave a little bit of space down here at the bottom. Otherwise, you'll want to, if you need more information about these nucleic acids, please go back to your Unit 1 mega sheet for some details. We're going to use this structure right here to drive a lot of our conversation for DNA replication. So hopefully this helps, and thanks for watching.